Hello there. I am delighted to be joined today by a CAR-T VIP, a pioneer in the field, Bruce Levine. Bruce is the Barbara and Edward Netter Professor in Cancer Gene Therapy at the University of Pennsylvania, past president and current chair of the Strategic Advisory Council of the International Society for Cell and Gene Therapy. Hi, Bruce.
Hi, Rob, nice to be with you. I just want to mention that I am the past president, so I'm no longer chairing the Strategic Advisory Committee, chairing the Committee on Ethics in Cell and Gene Therapy. So once you're with IACT, it's kind of an addiction to stay with them, and appreciate being able to speak with you today.
It's my pleasure. It's an honor, and I would like to know, how did all your current expertise get started? What first inspired your interest in science and medicine, and how did you then become involved in cell and gene therapies?
Yeah, so my father was a scientist, research scientist, and my mother was a nurse. So you could say growing up in a scientific medical household in the Apollo era, rockets going to the moon. And so I figured out pretty quickly that I wasn't going to be an astronaut, and astronomy involved a lot of math rather than just looking at the stars. So then set my sights on biology and kind of knew I didn't want to go to medical school and looking at graduate school, and went to the University of Pennsylvania undergrad and worked in a lab part-time to earn some money and got exposure to cell culture early. Following graduation, my father advised me take a job full-time working in the lab to make sure that's really what you want to do to go to graduate school. So I did. I took a technician position at the Children's Hospital of Philadelphia, and that was doing immune assessment on subjects that were enrolled in clinical trials of the varicella or chickenpox vaccine. And working in Children's Hospital, you see kids come in for their appointments. Some are in wheelchairs, some on stretchers, and that really drove home to me, why are you doing the research? It's to benefit people, and that sparked my interest in translational research. And so in grad school at Johns Hopkins, I purposely chose a laboratory that was in the hospital to do my thesis work. Again, you had to walk past patient wards to get to the laboratory. While doing my thesis work on T cell signaling, I read papers by Carl June and applied to his laboratory for a postdoctoral fellowship. So I had a background in cell culture, in immunology, and Carl put me on a project learning how to better grow T cells. And the result of that was a reagent that's used in research and clinical trials around the world to grow T cells. And pretty soon after that, he asked, how would I like to start and run a lab that manufactured T cells for clinical trial of adoptive immunotherapy? And it just seemed to me all that I had been interested in, all that I had been training for at that point, was a direct match with that ask. And so we started that lab in 1995 and began our clinical trial in 1996, and things just went on from there.
How did you get involved with the development of Kymriah?
Well, before Kymriah, it was a chimeric antigen receptor clinical trial that we first collaborated with a company called Cell Genesis, and this was in HIV. That was in the late 1990s. And when we moved to the University of Pennsylvania in 1999, we wanted to open clinical trials in cancer in addition to HIV. And Carl was very intent on continuing the chimeric antigen receptor work targeting antigens in cancer. So we got funding initially from philanthropy to start our preclinical work, and we called it CART19 for a CAR against CD19 in B cell malignancies. The first clinical trial opened in 2010, and we treated 3 patients, ran out of money because cell and gene therapy were not popular in the public imagination or in the minds of funders at that point, and decided to publish. Once we published those results, that got the attention of a lot of companies and people with money who wanted to license the technology. And that's how we came to have an alliance with Novartis. And so what we began as CART19 became CTLA-19 under Novartis, and then Tisagen, Leclusol, and the brand name Kymriah.
So can you remember how you felt when you realized that the therapy was going to work, and how did it feel to, begin to see the outcomes?
Well, to that point, we have been doing clinical trials and looking for evidence in terms of a immune response to HIV or to cancer, and maybe a rise in T cell counts and maybe some reduction in the tumor. And we were doing non-gene modified clinical trials. And so this was the first gene modified trial in cancer, the first CAR trial that we had done in cancer. And we were thinking, okay, maybe we'll see something, maybe we have to treat more patients, maybe we have to go to a higher dose or refine. And I'll never forget that summer and fall of 2010 and infusing the first patient. And there are samples that are taken in the days after to monitor for the T cell counts and the CAR T cell counts, and those were rising. And they're scheduled in the clinical trial at day 28 to bone marrow biopsy to look for leukemia. You look for it in the blood, but also in the bone marrow was most telling in terms of residual disease. And the bone marrow biopsy on the first patient came back, no evidence of leukemia. And I saw the email that went to Carl June from David Porter, and Carl said, I don't believe it because it was too good to be true. And so David Porter had to go back and say, well, the lab maybe made a mistake and we've got to get another sample. So a second bone marrow biopsy showed the same thing. And you think that is really remarkable, but maybe something happened we don't understand. We need to see what happens again. And then it happened again in the second patient, and then it happened again in the third patient. So we had 2 complete responses and 1 partial response out to 8 months in these patients with chronic lympholeukemia who were refractory to all available treatments. They had nothing left. And between 2.5 pounds and almost 8 pounds of leukemia were obliterated in the several weeks after infusion. So you think about re-engineering, teaching their own immune cells to fight back their cancer. That's a huge tumor burden to disappear over that time. And I'd say that fall of 2010 was the time when we knew that we had something that was really, really striking.
Okay, since Kymriah, how exactly have CAR T therapies advanced? And are we currently in a golden age of cell and gene therapy?
Yeah, I think we are, and we can advance beyond where we are. So there are currently 7 approved CAR T cell therapies. 5 are approved for acute lymphoblastic leukemia or lymphoma or chronic lymphocytic leukemia. 2 are approved for multiple myeloma. There are 4 approved CAR T cell therapies in China, 2 approved CAR T cell therapies in India, and 1 by hospital exemption in Spain. And that's not to mention the several hundred clinical trials investigating new approaches all around the world. So altogether, I think it's safe to say well over 50,000 patients have received CAR T cell therapies. Many of them respond, some of them don't respond, some of them relapse. So we have work to do to improve these therapies, not only in the indications where approvals have been granted, but beyond into solid tumors and other diseases.
What would you say were the biggest challenges in the field currently that yourself and your colleagues at the ISCT are focusing on?
Well, there are several big challenges. The technical challenges are scaling from research labs to clinical application approaches. And often there are challenges in doing that. There are what we call CMC, chemistry manufacturing control challenges that companies have faced in submissions to the regulators in defining potency for living drugs. And on the non technical side, I think we're focusing on the ethics of cell and gene therapy and communication and miscommunication in science. And that's everything from cell and gene therapy to vaccines and mRNA, which are used in cell and gene therapies, gene editing as well. So we've got to do a better job at communicating what we're doing and the value of what we're doing to our friends, neighbors, relatives, and ultimately legislators and those that are making funding decisions and investing decisions.
There is currently a lot of disruption, particularly in the regulatory department, and it's not just in the US, it's global. So what do you think can the CGT community do to help ensure science continues to thrive?
Yeah, so this is something that ICT has advocated for and taken on directly, which is we have new technologies that have developed over the past couple of decades, and they're unlike anything that many regulators have ever seen. So we need to provide education and training, not just to the scientists and professionals who are members of ICT, but also regulators. We bring regulators together. Every annual meeting, we have a global regulatory forum that is a closed door meeting to discuss current issues in the field, whether it's alternate approval pathways or rare disease or other issues. And we have another example is a course that we're running, a leadership cell and gene therapy course that is run by ISCT at the University of Pennsylvania, and we're going to stream it to the FDA. So we aim to provide opportunities to regulators to stay up to date on the latest science and to engage in alternative pathways while protecting patients. And we have another educational role that I mentioned, going back to the point on communication, which is there are shady actors that are direct marketing to the public on so-called stem cell therapies, exosome therapies, other therapies that have not undergone scientific and clinical testing that is appropriate to the composition. and they're marketing things for autism to Alzheimer's to orthopedic applications. So these unregulated therapies have the potential for financial and medical harm for patients, but also to harm the development of the field and confusing what is the scientifically and clinically validated pathway that is legitimate versus those making an end round on regulations and aim to make a buck.
Mm-hmm. That sounds like a great way to inspire the current generation of, legislators and regulators. But, despite all the disruption in that sector, how are you inspiring the next generation of scientists, particularly those interested in cell and gene therapies?
Well, I think it's reaching out and communicating like we're doing right now. It is all the activities of ICT from our annual meeting to our webinars and on down. And honestly, we've got to think creatively. One thing I've done that I guess was not done with so much advance intent is I co-wrote a song about CAR T cells that was recorded by a international country artist and released commercially in May with a music video. And we're running public service announcements on American radio stations to direct towards a website, ringthatbellsong.com, that has resources on CAR T cell therapy. So it's designed to call attention to the um, heroism of clinical trial patients and other patients facing a cancer diagnosis, the role of science and research, and as well as directing the general public to the website and some education around CAR T cell therapy. So, misinformation and disinformation is a huge threat to the field. It's a threat to the health and welfare of people worldwide. And we've really got to think creatively on, we can't just talk to each other. We can't just speak in scientific jargon. We have to learn how to communicate what it is that we're doing and the value of what we're doing. And the example that I use when I teach in a class or give a lecture is, how would you explain CRISPR or base editing to your neighbor, right? So being able to think about what you're doing, not just in the scientific context, but in the larger societal context.
Tell me about your new paper on innovations in CAR T cell immunotherapies for cancer.
Yeah, what we wanted to do here was not just to write another review on CAR T cell therapies, of which there are many, but to really focus on the future. And to that end, we picked three areas of particular interest that are going to advance the potency, the accessibility of CAR T cell therapies. And the first of those is, we'll call it armoring. And this is addressing a challenge that we find in solid tumors and cancer in general, which is cancers have evolved ways to hide from and suppress the immune system. So how can we engineer T cells to be resistant to those effects of the tumor? We call that armoring. One example is to incorporate an anti-tumor cytokine into the CAR construct, and we've published on that, integrating IL-18 recently. Another section deals with genetic enhancement with the discovery and implementation of CRISPR as a tool. There have been CRISPR screens of CAR T cells and T cells to look at what are the genes that are upregulated, downregulated, what can increase function, decrease function. So there's a long list of gene targets that you can use to enhance function, and this is applicable to other immune cells and likely a subset of those to other cell therapies in general. So that's the genetic enhancement section. And then finally, we have those approved CAR T cell therapies. We have approved cell therapies. Those therapies rely on removing cells from a patient by leukapheresis, growing those cells up in the lab, genetically modifying them in a lab, shipping them back after doing quality control assays. It's a very laborious and expensive process. And when we have approvals in solid tumors, when we have approvals in autoimmune disease, other diseases, you can multiply by 10, by 20, and more the number of patients that could potentially benefit. And I will say pretty confidently there aren't enough humans on Earth, there aren't enough reagents, at least at present, to be able to serve all of those patients. So that third section deals with the in vivo production of CAR T cells. And the means there is by direct injection of a viral vector encoding a CAR or a lipid nanoparticle that encapsulates messenger RNA. And in that case, you're not making the therapy in the lab, you're delivering a reagent that genetically modifies T cells in the body, and then that creates the CAR T cells that can have that clinical effect. So those are the three sections of the paper, and we're excited to see it in cytotherapy along with the whole other collection of papers in that issue.
Yeah, it certainly seems very promising. But where else do you think future research priorities might lie?
Well, we have a challenge, I think, in the U.S. and UK and in some other regions of the world, which is research and development and science funding. So there's never been more excitement around the possibilities of developing therapeutics, but there's also never been more uncertainty on the funding of science in countries around the world. So this is our mission at ICT and in the other professional organizations at the universities, which is to focus on our work, of course, and submitting grants and recruiting philanthropy, industry investment, but also the importance of communication of what we're doing. Ultimately, we aim to treat disease and save lives, and we have to communicate that value to the public.
Bruce Levine, thank you very much.
Thank you.